measures are also called vaccine efficacy. So in vaccine trials, when you see the word vaccine efficacy, it means relative risk reduction. What's not reported often are the absolute risk reductions. And that's important because that, absolute risk reductions are more relevant to clinical needs and purposes and applications, to public health applications, whereas relative risk reductions are used for research purposes by researchers. So the relative risk reductions by themselves are not going to give you the information that people, the public, or the physicians, clinicians, or even the public health authorities need to decide, well, how effective or efficacious is this treatment really? And there can be a wide difference between the relative risk reduction and the absolute risk reduction in the same trial. Now, people need to hear both those numbers in order to come to the proper conclusion about the efficacy of the treatment. If you're only getting one number, you're missing the important information, and that could mislead you. And this is a common problem in research today. Many healthcare professionals and the public don't understand the difference between these two measures.
Now, does the FDA require absolute risk reduction? And if so, why do companies rarely use it?
Well, you know, this is interesting because the FDA has published guidelines saying that when research information is disseminated to the public, communicated to the public, absolute risk reductions should be included with relative risk reductions. It's there right in their guidelines because they understand the problem that I was just trying to describe. Now I can get into the details as to why the calculation of the absolute risk reduction is so much different than the relative risk reduction. And to do that, I would have to get into a little bit more detail to describe exactly how a randomized trial for a vaccine is set up. Who's in which group, and how do we count the infections and the efficacy of the vaccine?
Absolutely. Please go ahead.
Well, there's a figure I have in one of my publications that shows you just this. Let me get it up here so I have something to guide myself with. Okay, so in this figure, which you can post while people are listening to my voice, we see two groups, a vaccine group and a placebo group, and we see that there are 100 people in each group represented by these gray figures. And among these gray figures are these red figures called events. In this case, the event is an infection incidence. So in this case, the infection is SARS-CoV-2. More specifically, for the COVID-19 vaccines, they're infections that are symptomatic. So there's at least one clinical symptom with the infection. Now, incidence means that all of the people in the group started out without an infection. So as during the course of the trial, as one person develops an infection, that's known as an incidence. So what we do is we calculate and record the incidence rate of the infection, the event that we're looking at in each of the groups, the vaccine group and the placebo group. Now, it turns out that the vaccine group has another name. It's called the experimental group. And so the rate of the incident in that vaccine group is called the experimental event rate. And the same for the placebo group. The placebo group is called the control group. And so the events that occur in that group are called the control event rate. So the control event rate is the acronym is CER. The experimental event rate is EER. Now, I realize I'm using a little bit of jargon here, but that's why I want you to look at the figure so you can refer back to this and study it a little bit more so that this information becomes more familiar to you. So in the example that we see in this figure, in the vaccine group, we have one red event, one infection incidence. So we have 1 out of 100 people. That's a 1% risk of the infection. We have an experimental event rate, an EER of 1%. In the placebo group, we have 2 infection incidences, 2 events. So that means we have a 2% risk. The control event rate, the CER, is 2%. So now we know the EER, the experimental event rate, is 1%, and the control event rate, the CER, is 2%. Now we can calculate our absolute risk reduction and our relative risk reduction. And you see that on the right side of the figure. The absolute risk reduction is the vaccine risk reduction of the disease. And we calculate that by subtracting the EER, the experimental rate in the vaccine group, from the CER, the control event rate in the placebo group. That gives us the ARR, which is the absolute risk reduction. So in this example, we're subtracting the 1% experimental event rate in the vaccine group from the 2% control event rate, the CER. So 1% subtracted from 2%, it equals 1%. There's your absolute risk reduction. So this vaccine, in this particular example, was effective for clinical purposes to reduce your risk of an event, the infection, by 1%. Now, that doesn't sound too impressive until we consider the relative risk reduction. We use the same exact numbers to calculate the relative risk reduction. The relative risk reduction is supposed to represent the vaccine risk reduction relative to the group that didn't get the vaccine. That's the whole idea behind it. So if we know the reduction in the infection in the vaccine group, which is the ARR, the absolute risk reduction, which we just calculated, we divide that by, in other words, it's relative to the placebo group and the risk in the placebo group, or the CER, the control event rate. So in other words, the absolute risk reduction, as you can, and all this is on the figure, so you can follow along as I'm saying this. We take the absolute risk reduction, which we already calculated is 1%, divided by the CER, the control event rate, of 2%, and that gives us a relative risk reduction of 50%. 50% compared to an absolute risk reduction of 1%. This shows you the power of the relative risk reduction. And it shows you how, if you only had that relative risk reduction number to determine how effective, or whether or not you wanted to use this treatment, without the absolute risk reduction, you would come to, let's face it, a biased conclusion. And I call this an outcome reporting bias. It's biased because you're not getting both numbers, which, by the way, as you recall, I already said the FDA had recommended needed to be conveyed to people when you're disseminating this information to the public. So why have the relative risk reduction at all? The reason we have that is because it makes a good way to quickly summarize in this case, the vaccine efficacy between different trials. That's useful for researchers who are quickly want a comparison between trials. However, it is not useful for clinicians and the public health experts and politicians and the people in general who all need to determine how effective this will be in reality for reducing their risk. In that case, the relative risk reduction is not effective. It doesn't mean that you shouldn't disseminate that information, but you have to also include the absolute risk reduction.